All right, so you all had your first quiz last week, and it was just on the general topic of blood. And then we have this particular question that I had asked about that, and this is going to be typical for the quizzes that I give you where there's a lot packed into a single question. And the trade-off that we have going on between us is that you have multiple chances to show me that you understand blood. So you won't see this exact question again, but as you build your understanding of this question, you will build your understanding of blood generally, and the next time you take the blood quiz, it won't be exactly about this, but you'll probably have more knowledge at that point. So I'm going to take you through how I think about this question. I'm not going to necessarily give you all the answers because I kind of want you to just get a little bit of input from me on it, revisit it, go back into your text, back into the videos, start to kind of build your understanding to the point where you have a functioning picture in your head. You're going to see me do a lot of drawing. I'm going to emphasize that a lot. Because if you can draw how you picture something in your head, it means you actually have a sense of it, an image of it in your head. If you just kind of use words that you've heard before, it's very easy to not actually grasp what's going on and just kind of have a superficial familiarity with the words. All right, so we're going to take this case. We've got a patient with type B positive blood, and they need a transfusion of plasma specifically. So they're not getting a whole blood transfusion. We're just going to do that trick where you can spin the blood and get the plasma fraction of it. And that is very useful, especially for people with hemophilias and bleeding disorders, because the plasma is where all your clotting factors are. So you don't have to give the person whole blood, you just give them the part of the blood they need for their hemophilia, because they need those clotting factors in order to be able to not bleed to death. So there's this medical student, and the medical student says the patient only needs plasma, so since we're not transfusing whole blood with all the cells in it, we don't have to worry about matching blood types. So the question for you all was to figure out, is that true? If you only give plasma to a patient, are you kind of free of having to worry about a mismatch in that transfusion? So first step here, we're going to start with this kind of type B positive blood thing. What I want you to do is for any blood type I give you, I want you to be able to draw our little cartoon red blood cell, right? And we know that on the surface of those red blood cells, we're going to have different kinds of proteins. They have different words. Sometimes they're called antigens. Sometimes they're called agglutinogens. But what they are is little pieces of protein that stick up off the surface of red blood cells. Some of us have them, some of us don't. Some of them have some, but not others. So what we're focused on here is if this is going to be a B positive red blood cell, we have to figure out, does this patient, who is B positive blood type, have any antigens sticking up off the surface of the cell? Some people have none. So is that the case with this patient? So we have two different factors here. We've got the ABO blood group, so the B part, and then we've got the positive part. So those are two different pieces of information about different kinds of antigens that could be on this red blood cell. So since we have type B blood, we know that one type of protein that's going to be sticking up off the top of these red blood cells are B antigens. So that's what that B part of the blood type tells us, right? Now the plus part is telling us, I'll just switch colors just so that we can keep this kind of straight. The plus is talking about that rhesus factor antigen. So if you are a positive blood type, that means you do have rhesus antigens. So I'm going to put rhesus there. Yep, rhesus. I'm going to make these flags bigger so I can see what I'm doing. All right, so that's what a B positive red blood cell looks like. It has two different kinds of antigens sticking up off of its surface. And then we're going to look at what consequences that has for blood typing. So as a check your understanding kind of thing, what I want you to do, so I'll give you a couple of examples to practice on. Let's say that you have been told that a patient is AB negative blood type. What I want you to think about as a little check, and you can always talk to me or check your answer by Googling it, what antigens are on this red blood cell? So that's a good check for you at the first phase of this. Versus, okay, let's take a different patient. This patient is going to be O negative. Same question. What antigens should you draw on the surface of that red blood cell? So that's our first step, is to be able to consistently do that. And then the other thing I want you to be able to do is, let's say I give you a red blood cell, right? And I'm going to change colors. Let's say I give you a red blood cell and I show you a little cartoon of it, and it says A. So this is what the red blood cell looks like. Then what I want you to be able to do is tell me what blood type is this patient. So that's the first phase of this whole blood typing thing. You should be able to do this in both directions. If you're given a picture of a red blood cell, kind of a cartoon of one, be able to tell, just using that picture, what is that person's blood type. And then the flip side of that, check your understanding, is if I give you a blood type, AB negative, O negative, B positive, any of those, you should be able to draw a cartoon red blood cell and show whether any antigens would be on its surface, and if there are antigens on its surface, which ones would be there, all right? So that's the first thing. We have a patient with B positive blood, so we need to make sure we really understand what B positive blood means. Then we've got this second part of the question that's talking about plasma. So a lot of you in your quizzes were kind of like sort of trying to figure out like, well, is this medical student onto something or not? Because we've been really focused on the red blood cells themselves when we talk about blood types. But now we're going to get into the plasma part of things. So if we kind of zoom in, we've spent a little time looking at these blood samples that we spin down so we can separate the components. And when you spin down blood, the cells all settle towards the bottom and the plasma stays up at the top, right? So we know that plasma is mostly water, and that's what keeps your blood flowing. All the cells are floating around in this kind of watery plasma, but there's other stuff in the plasma. And what we're going to focus on is this component, this protein component, because that's what we're kind of interested in. So water, not a problem. We can transfuse water into a person. We're not too concerned that water is going to cause an issue necessarily. But what we're wondering about is, okay, plasma contains all these different kinds of proteins. For our patient, is there any possibility that when we transfuse in this plasma donation that our patient needs, that there will be a bad reaction between her cells and that plasma coming in? So a lot of you rightly pointed out that the plasma doesn't have any cells in it, right? So once you spin down the blood, this plasma part is acellular, so there are no cells in it. So the question is, does that make it safe to transfuse into a patient where their blood types are mismatched? Because a lot of you pointed out, well, there's no red blood cells in plasma, so there shouldn't be a problem. So that's kind of what I want to focus on is that where the conflict would come in? Is that why a patient would suffer if you gave them a mismatched transfusion? So what I want you to be able to do, I'm going to try and change this. So let's look at a blood vessel, all right? So here's the walls of the blood vessel. I like this. It's kind of like a little watercolor tool. All right, so then if we have red blood cells, so here's our patient, right? Okay, so here's her red blood cells. This is what's floating around in her system right now, all right? And then we know since she's B positive, we figured out she's got little B antigens all over her red blood cells. So that's sitting on all of these guys. And she's B positive, so she's also got rhesus factor sitting on top on the surface of her red blood cells. So this is what her red blood cells look like in cartoon form, right? So now we know those red blood cells are floating around in the plasma. So this is her own plasma. This is the patient's plasma. And we know plasma is mostly water, but it has a bunch of proteins in it. The proteins we're going to focus on are the antibodies. Antibodies are a type of protein. So we know that's one of the things that's floating around in her plasma. Now, antibodies can come in different shapes. Sometimes they're shaped like little, kind of Y-shaped sort of things that look like this. So this is one kind of antibody. There's others that are much more complicated looking that look like this. So they have like a 5 kind of, almost like a starfish shape. So this is another kind of antibody. All right, so they can come in multiple shapes. They actually can come in double-ended kinds as well. So these are all antibodies, all right? So this is antibodies, right? So this is like what all these guys are. Now, antibodies are what your immune system uses to flag or tag, sorry, these notifications should not be happening, to flag or tag things that don't belong in the body, that are not part of the self, that are not part of that patient's body. So what that means is that for our patient here, B antigens and rhesus antigens are self. So B and rhesus equals self antigens. So her immune system should be really chill if it detects a B antigen or a rhesus antigen, because they belong there. They're part of her body. They're not anything that's going to work up the immune system. The immune system ignores them. It does not flag and tag them because they are supposed to be there. Now, if you introduce an antigen into her body that is not supposed to be there, right, it's non-self, her immune system, so these antibodies here, are going to detect that antigen and flag it and tag it, all right? So what we have to think about is what types of antibodies would she have in her system? And then we have to think about, okay, if we're going to give her a transfusion of somebody else's plasma, what might be coming in from there? So if we look at her antibodies, one question that, I might phrase for you guys is, all right, so I'm going to phrase it this way. What antibodies, so shorthand for antibody is AB, what antibodies would be in her plasma? So antibodies generally come in flavors, right? So you can have anti-A, anti-B, and anti-rhesus, right? So the question for you to answer, just kind of pause and think about this before you proceed in watching the video. Which of those would be present in her plasma? Would she have anti-A antibodies? Would she have anti-B antibodies? Would she have anti-rhesus antibodies, right? So what we want to think about here, after you've thought about that for a little bit, we want to kind of picture the conflict here, right? So if we've got a red blood cell here, right, and it has, just really digging this kind of paintbrush tool, if we have B antigens sitting on the surface of this cell, right? Now, the problem that's going to happen, I'll draw you an illustration. Here's another one of her red blood cells. So we want to look at what is the nature of the conflict that could happen in a body where there's a mismatch, okay? So we want to remember she also has rhesus antigens. So I'm going to put those guys here. All right. Now, what we're concerned about here is a mismatched transfusion where the red blood cells are going to be flagged and tagged for destruction. And when they get flagged and tagged, they're going to start sticking together. So I'm going to show you an example of how this ends up looking. So here's this guy. Here's this guy. So remember those kind of starfish-shaped antibodies that I drew for you? Here's one of those guys. Doop, doop, doop. They look like that. So what's going to happen is this. So this right here, this is an anti-B antibody. What that means is this particular antibody is programmed to recognize B antigens wherever it finds them, and it will latch onto them with this kind of claw part of the antibody. So right here, we've got one arm of this antibody has discovered this B antigen. And then because there are 5 arms on this antibody, another one of these claws can latch onto a neighboring red blood cell that also has a B antigen. So now what has happened is that instead of these 2 red blood cells being free to kind of like smoothly flow through the bloodstream separately, they're now stuck together because this antibody here has connected them to each other. So we've got these 2 arms are kind of velcroing these 2 red blood cells together. And this can keep happening. So because this antibody has 5 arms, there's another spot here where it could find a B antigen and latch onto it. So you could end up with 5 different red blood cells all now stuck together. And that is a clump, and clumps can lead to clots, and clots can lead to death. So we don't want this happening. We don't want antibodies running around the system detecting the patient's own red blood cells and making them clump together. That is what we want to avoid. So if we think that in our patient, transfusing mismatched plasma into her might cause a problem, then we don't want to do it. We don't want to lead to clumping, right? So if we proceed, and hopefully you kind of took some time to figure out what might be in her system, right? So we've got her bloodstream, okay? What's in her bloodstream? A bunch of red blood cells, right? And we've got B's and rhesus all over her red blood cells. Rhesus, B, rhesus, B, rhesus. Okay, so what this means in terms of the antibodies that would be floating around in her body, there better not be any antibodies against B antigens. There better not be any antibodies in her circulation against rhesus antigens. Because if there were, then her blood would be clumping all the time. So if we were able to zoom into her plasma and see what kinds of antibodies are circulating in there, we would find some. So I want to leave more questions here for you to pursue, and then we can kind of talk about them as a group. What kinds of antibodies are normally present in her system? Okay, so what kind of antibody are normally present in her system? All right, so thinking about that, and sometimes you have these little double-sided antibodies. So this is kind of like looking at, if we were to think about what's circulating in her plasma normally, before she's gotten any transfusion, what kinds of antibodies would you expect to find in her normally, and what kinds of antibodies would you not expect to find in her normally? So now I want us to think about this plasma transfusion, right? So we know plasma is where the antibodies are found. So if you're going to transfuse, so I'm going to put like this big kind of sharp needle coming in here, and I'm just going to try to erase this and show this needle coming in, and we're going to say, okay, so now there's going to be some plasma coming in from a donor, right? So we've got plasma, this is donor plasma coming in through the needle. It's going to enter her body. So this is donor plasma. All right, so what I want you to think about here is, let's say the donor plasma is blood type A negative. Is this a problem? That's basically what I want you to think about. So I want to leave this with a little space for you to reconsider your answer to the quiz, see if you want to elaborate on it, change it, anything like that. What I want you to think about, based on what I've shown you here, is we're going to think about this donor who is a type A negative blood, all right? We want to remember we're going to spin down that donor blood. We're going to take just the plasma part. Right? So we're not going to transfuse any red blood cells from our A negative donor, but we are going to transfuse in whatever is in that plasma from that type A negative donor. Now that we've kind of re-looked at the fact that the plasma is where the antibodies are, what I want you to think about is what type of antibodies would be in an A negative donor's plasma. And then the next step is to think about, okay, if I take that plasma and I put it into a B positive patient, what do I expect will happen? Knowing that whatever antibodies are circulating in that A negative person, they're now going to be entering the bloodstream of our patient who is B positive. So now, given that, I want you to take it to the next step and start kind of diagramming out what would be sort of the cartoon visual of what would happen there. Once you've drawn that out, we can talk about that. You can kind of show me what you think of that, and we will proceed from there. But I really want to pause, and rather than giving you the answer on this, I want you to kind of wrestle with it a little bit. This is sort of, challenging yourself, seeing how much you can make of it, and really thinking through. There's multiple layers here. You have to be able to picture the patient's blood. You have to be able to picture her red blood cells, what antigens are on those red blood cells. Then given that, what antibodies would be normally circulating in her system. And then the next level is, okay, if I now give somebody else's plasma into her bloodstream, is there going to be any kind of conflict? The bottom line here is what we're worried about is that there will be an interaction between antibodies that are attacking the antigens on somebody's red blood cells. So there's this back and forth here that can cause a little hiccup in your brain where you're trying to keep straight, okay, it's the antigens, they're on the red blood cells, what's attacking what would be the risk here. So I'm going to stop this here and let you just kind of think about that. Refer back to your textbook on this, refer to the crash course video that I posted in Blackboard to kind of, guide you through some visuals on this. And then we can look at this again and see if we can come to some, further understanding, better understanding of exactly what this ABO rhesus transfusion mismatch whole thing is.